Hi, I'm Sharon Cantor from the University of Massachusetts Chan Medical School, and I'm happy to be one of the co-organizers for the DNA Replication Gap Cancer and Disease meeting that will be happening in Daejeon, South Korea, April 27th through 30th in the year 2025. My vision for this meeting was to bring together the brightest minds in this emerging single-stranded DNA gap field. Gaps represent a transformative paradigm shift in understanding of cancer biology, therapy resistance, and even aging. And so I think the field, like me, is just really curious to know how far this gap biology will go. Yeah, this one stands out because it's really the first public forum entirely dedicated to the DNA gaps. So historically, gaps have been overlooked in favor of DNA double-stranded breaks. But gaps have really revealed themselves as being critical determinants of chemotherapy response and really provide new opportunities for biomarker development and therapeutic targeting. So beyond cancer, gaps also are likely to influence cell fate decisions, stem cell attrition, genome stability, and they can link them to aging and developmental disease. So the groundwork for this gap meeting has been laid with foundational papers, but this is the first public forum dedicated to this topic. So by, sort of having this hub, for cutting edge research happening right in South Korea, we'll be able to bring together all these excellent labs that are emerging, across Asia and in China. And that connection, again, has been really limited. And lots of meetings have been canceled or postponed. And this will allow us to now, sort of create a vital link between these growing research communities. I think, this meeting will bring together the most sort of unparalleled opportunity just to completely explore this really new area of replication gaps. And so, from like how they form, how they're repaired, how to best visualize them, what are their structures using cryo-EM, super resolution imaging, cutting edge technologies like nanopore sequencing, what is the architecture of these gaps and what proteins are interacting with them, ultimately linking gaps to therapy response and resistance. We have all these emerging DNA damage response inhibitors coming online in addition to PARP inhibitors, Pol theta inhibitors, CDK inhibitors. How do these drugs influence gaps and ultimately confer their therapy response? So one of the aspects of this meeting that I'm excited to explore is, DNA gaps beyond the replication context. So this is particularly relevant for understanding post-mitotic tissues like brain, where increased mutagenesis is not fully explainable by DNA replication stress. So it's emerging evidence that NICs may drive mutagenesis in these contexts. And so this offers a new perspective on the underpinnings of neurodegenerative diseases. At this point, we're sort of stuck with these very slow, laborious, error-prone assays to study replication gaps. And so what we're excited to share with the field coming online are different kinds of assays which are going to really just democratize this field, and that other people who haven't even entered here yet can now study replication gaps in their labs and then start to ask their own questions. You get trainees to very established investigators coming together, talking about this field in a sort of holistic manner that we can start to be inspired about which directions we can go and what sort of parts of the field we need to emphasize and gain more opportunity. If you can submit your abstract by January 28th, 2025, you'll be able to be considered for a short talk. And if you can't make that deadline, that's okay. If you get your work in by April 3rd, 2025, now you can be eligible for a poster abstract. And that also ensures that your work will be featured at the meeting and get to interact with lots of people at that time. So we hope you'll join us to learn about these techniques and how we can move this field forward together.